Okay, we're going to get started here for the next speaker. I'd like to introduce Dr. Gary Ulaner he's a clinical professor of radiology and cancer biology, and he is the James and Pamela Musi Endowed Chair of Molecular Imaging and Therapy at the Hoag Cancer Institute and a professor of radiology and translational genomics at the University of Southern California. He's also on the editorial board of 4 leading radiology journals, and this week he was named as SNMMI's Vice President-elect. So he's kind of bored. He doesn't have a lot of spare time. So congratulations, Dr. Ulaner, and it's our pleasure to welcome you and look forward to your talk. Thank you.
Thank you, Mike, and thank you for being here. My name is Gary Ulaner, and I'll talk about clinical trials, how to understand them, evaluate them, and find where they are to see if they would be appropriate for you. I am in wonderful Southern California at the Hoag Family Cancer Institute and the University of Southern California, and I have to say, walking outside from here in New Orleans and my glasses steam up is kind of a new experience. It's something a little new. These are my disclosures. More importantly, I disclose that I like to talk about molecular imaging and therapy starting at a very early age. This is my son, Ilya, and my daughter, Annabelle, who helped me teach a molecular imaging and therapy course in Southern California every year. And you can see, if you do train them early, that they get very good at finding the important things, especially my daughter, Annabelle. If anyone is looking for junior faculty in molecular imaging and therapy, I have people to send your way. So what are clinical trials? At heart, clinical trials are studies that are performed to test new medical treatments or procedures in humans. They are the primary way how we can determine if a new treatment is both safe and effective in people. So you need to demonstrate both safety and efficacy in order to become approved by the U.S. Food and Drug Administration, which is why in most eras the U.S. FDA is so valuable in helping us choose the best treatments for patients, because we have this incredible data which shows, yes, what we're using is both safe and effective, and if it doesn't reach both of those criteria, then it doesn't get FDA approved, right? A difference between medications versus some other alternative therapies that are available. While I am a proponent of alternative therapies for some indications, the alternative therapies do not have a central stamp from an organization that says, yes, we are both safe and effective. So what are the types of clinical trials that are available? There are essentially these 4 different types of trials. Diagnostic trials are ones that test new ways of diagnosing a disease but not treating the disease. These would be in nuclear medicine, these would be our radionuclide tracers that try and detect where is the prostate cancer, where is the neuroendocrine tumor, where is the breast cancer, but we're not attempting to treat the disease, just to discover a better way of detecting where is the disease, how much of the disease is there, and is the disease responding to treatment or not, right? The second, I'd say for diagnostic trials, these safety is not, they have very few side effects, these diagnostic agents. They're used in such small quantities that they usually have little to no safety issues. And then it's really efficacy. Are they really better than what we had yesterday in order to determine whether it gets FDA approved? Treatment trials are trials that test new treatments for a disease. So they're giving a new agent, and then there are downstream measurements: is the disease decreasing, getting better over time? These medications definitely do have more safety concerns, right? It is unfortunate, but we are unfortunately imperfect. We do the best we can, but virtually every treatment that we have available for cancer unfortunately has side effects, and you have to balance those side effects, the safety, with the amount of benefit, the efficacy that you have in order to determine whether it's worthwhile for you to undergo a treatment. Prevention trials are looking for better ways of having to prevent a disease from occurring in the first place. So for example, there is a spectrum of diseases called plasmacytomas. The disease, the real bad disease, is multiple myeloma. There are precursors of multiple myeloma, like smoldering myeloma, and there are interventions that are geared towards how can we prevent the smoldering myeloma from becoming multiple myeloma so people don't get the adverse effects of multiple myeloma. Those are prevention trials. And finally, behavioral trials are ways that we try and change our interventions that try and promote behavior changes which lead to better health. And I think obviously the best example of those are smoking cessation, right? If we can get people to not smoke cigarettes, you get rid of 1/3 of all chronic disease, 1/3 of all heart disease, 1/3 of all cancer. So while I don't personally work on behavioral trials, I applaud the people who are doing this work, because if we can get the behavioral trials to help people omit these things that cause disease, well, there'll be less need for people like me who have to treat disease. Next, what are the phases of clinical trials? You've probably heard of the phases of clinical trials, and they go from 0 to 4. Most people are probably most familiar with 1 to 3. 0 is essentially, and it increases, right? Zeros come before 1s, 2s, 3s to 4. Zeros are really about gathering preliminary data in order to lead to a phase 1 trial. Many agents do not have phase 0 studies performed on them, but sometimes phase 0s are performed. So if you're asked to consider a phase 0 study, this is very early. That's the earliest data collection on a particular agent. Phase 1 trials are official trials that test safety in a small group of people. Now, efficacy would be wonderful, but just be aware that the major goal of phase 1 trials are determining safety. So they tend to start at a low amount, what we call dose of an agent, and then when safety is achieved at the first dose level, they give a little more to the next group of participants in a clinical trial. And if that level is determined to be safe, they give a little more. Now, this is going to sound harsh, but it's true. These type of phase 1 trials are designed to try and increase dose until you hit what we call adverse events, right? So that we know that when we move forward on a clinical trial, we're doing it at a dose of the medication that will not cause severe adverse events in the majority of patients. So it is not uncommon, as the dose is being escalated, to hit a level where there are too many side effects. That is one of the disadvantages of a phase 1 trial. You could be in that cohort. The major advantage of a phase 1 trial is it gets you access to a medication potentially years or a decade before other patients can have access to that medicine. So for things that are really particularly very promising, phase 1 trials can be remarkable successes for patients, but you just have to keep in mind that phase one trials, physicians still aren't sure what is the appropriate amount of the medicine to give you in order to optimize efficacy versus adverse effects. Phase 2 trials are next and are larger trials that are looking for what we call signal of efficacy. Okay? We want a signal that something works. We want to see that something will work in a group of patients before going on to these really large phase 3 trials. If we see no signal of efficacy in the phase 2 trials, then they don't move on to phase 3. So again, phase 1, safety. Phase 2, efficacy. Phase 3 tend to be these more randomized trials where they're comparing the new medicine versus what was previously available to see if what is being proposed is an incremental benefit over what was already available previously. Phase 3 trials are quite large. They're usually performed at many different places around either the United States or the world, and they compare the efficacy and safety of the new medicine, often against a medicine that came before. All right? That's our 1, 2, and 3. The phase 3 data is usually what is presented to the FDA, the randomized data, and then the FDA reviews, hey, is this medicine safe enough and efficacious enough that it should be approved, and then physicians are allowed to prescribe it off of a clinical trial. Right? So again, in the United States, if I were to prescribe a medication, what I prescribe either has to be FDA approved or on an FDA approved clinical trial. I can't prescribe medicines that don't meet one of those 2 criteria. Different countries have different rules. So sometimes other countries you can get access to a medication earlier because they don't have that rule. They have rules like compassionate use, which says that any doctor can prescribe any medication that they want if they feel it's beneficial for their patient. That has advantages and disadvantages. Again, advantages, it gets treatments to you quicker. Some countries had Lutathera and the agent in Lutathera, the lutetium-177 dotatate, and then the agent in Pluvicto, the lutetium-177 PSMA-617. They had them earlier than what was available widespread for patients in the United States because they had this compassionate use criteria. But also, you have the downside that those countries were also using things like thalidomide and causing birth defects in large numbers of patients. So you get advantages and disadvantages. When you're in a location that has the more compassionate use, you can get it earlier, but there's more risk of problems with safety efficacy. And when you're in a place like the United States that you have to have FDA approval of the drug or the clinical trial, you may get it a little later, but the US FDA really is right now the gold standard in the world. For many other countries, they wait till the US FDA approves it for other countries to have widespread use of it because they trust the US FDA's safety and efficacy evaluations. Phase 4 trials, and I'm a big fan of phase 4 trials, and we run several of them, are after FDA approval. So you think, why would I need something after FDA approval? Well, often it is for longer-term study of that safety and efficacy, or for determining a new indication for the medicine. So the VISION trial led to FDA approval of lutetium-177, PSMA-617, what we call Pluvicto, for patients with metastatic prostate cancer that had hormonal and taxane chemotherapy previously. There was a trial there are now trials that are bringing these PSMA agents earlier into the course of the disease, and some of these can be considered phase 4 trials post-FDA approval because they're trying to expand the patient populations that can benefit from the medication. Okay, so why participate in clinical trials? Well, to the one that I think most people state is that clinical trials allow you access to potentially efficacious medicines years before other people have access to them. So a lot of people have been, particularly I think we're fortunate in nuclear medicine with radioligand therapies, patients have wanted to get on our radioligand therapy trials because the medicines as a class have proven to be more efficacious and have fewer side effects than many chemotherapies for patients with prostate cancer and neuroendocrine tumors. So now there is great interest in patients with other malignancies to get onto these trials with the hope that medicines for patients with breast cancer, lung cancer, pancreatic cancer, and other malignancies will have similar benefits in efficacy and safety. But beyond that, the second bullet point here, if you're on a clinical trial, you get more attention and more time is spent on your care than if you're not on a clinical trial. All right? So there's no hard data on it, but I would think having more people having eyes on you at more points of time mean that there's more opportunities to evaluate when are things going well and when are things not going so well so that you can change to things that may be going well. So I've always been a big proponent of clinical trials, as even if the medicine turns out not to work, you have more people looking at you more often, which gets your concerns answered quicker. And I truly believe that being on a clinical trial, overall, you get better care than otherwise. The third point is more altruism. Being part of clinical trials helps develop the medications of the future. So in addition to possibly improving your own health, you are contributing to the advancement of medicine and science for people in the future that benefit. If you are using Lutathera or if you are using Pluvicto, there are a plethora of patients, doctors, nurses, coordinators, etc., that had to participate in the trials that led to the FDA approval of that agent before other people now can use it on standard of care, and I personally consider that a valuable benefit as well. So I think there are both personal and altruistic benefits to being part of clinical trials. So I didn't see Esma's presentation, which I'm sure was wonderful, but this is my cartoon of how molecular imaging and therapy trials work, if this hasn't been shown before. I like to say, and I do oncology work, so I'm going to talk about cancer, but this can apply to different diseases. We start with a cancer cell. We identify a target either on or in the cancer cell. We then design a binding agent that binds to that target, much like a key fitting into a lock. So if my cancer cell has the target, my agent hits it. If my cancer cell doesn't have the target, my agent doesn't hit it. And hopefully, most normal cells don't have our target, so normal cells are spared. We then link, there's a linker, link a radioisotope to our binding agent. And this creates these keys that we use for imaging and therapy. If the radioisotope that's linked to the binding agent emits a small amount of radiation, for example, a positron, that's an imaging agent. It's something that I can administer into the bloodstream of a patient. It travels everywhere in the body where their blood goes, and cancers need blood in order to get nutrients for them to grow, so cancers always get blood. So if I put it into your bloodstream, if there's cancer anywhere in your body, we have the opportunity of getting to it. And then again, if the target is expressed, our key binds to the cancer cell, and the emission of the positron, the low amount of energy, allows us to detect where and how much cancer there is from head to toe. Now, for therapy, I remove the radioisotope that emits a small amount of radiation, and we replace it with something that emits a lot of radiation. These are the beta particles, the alpha particles. We talk about Auger electrons. These are things that emit enough radiation that we can kill cancer cells. And again, now I have a key that can be administered intravenously. It circulates all around the body, wherever the blood flow goes, and has the opportunity of binding to every single cancer site in the body. And this system, I think, is so powerful because we have the imaging test before the therapy. So most treatments, like chemotherapy, they just give you the therapy, and then they wait 3, 6 months. How long are they going to wait till they measure whether the medicine works or not? Well, what molecular imaging and therapy does is you do the imaging first to see whether you have the target. If you don't have the target, you don't do the therapy. And by doing this, we can separate out the people who are not going to respond to that therapy and don't waste time, don't waste side effects on therapies that we know are not going to work, and we enrich the number of patients that are likely to respond to the therapy. All right? Here are examples. Right? I think the first of the example of this is for neuroendocrine tumors. We have where the cancer cell is the neuroendocrine cancer cell. The target are the somatostatin receptors. We have a binding agent called TATE, which is an amino acid structure. The linker is called DOTA, and thus we have DOTA-TATE. We link to the DOTA gallium-68, which emits a positron, and I have a key, which is an imaging agent. If I link lutetium-177 or actinium-225 or some other high-energy emitting radioisotope to the DOTA, I have a therapy agent. And I can do the imaging agent first, make sure that someone has the target, and then do the therapy agent. And this, I'm sure people have seen, is the success of both imaging and therapy with radio-labeled DOTA-TATE for patients with neuroendocrine tumors, where first we can see the disease with the imaging agent far better than was previously possible. And when we look at the results of the NETTER-1 trial comparing the therapy agent outcomes with prior standard of care, there was a remarkable improvement in progression-free, and then a less impressive but still remarkable improvement in overall survival. I think this launched the field of molecular imaging and therapy as we know it today. And this was now for neuroendocrine tumors, we are replacing what's bound to the DOTA with more potent emitters of radiation like actinium-225, which is an alpha emitter. And this is an ongoing clinical trial to see if the actinium DOTA-TATE can provide benefit for patients who have benefited previously from the lutetium dotatate, which is Lutathera. All right. Let's move on to trials, molecular imaging for prostate cancer. There's a whole lot more. All right, so it started with neuroendocrine tumor, but for prostate cancer, now the 2/3 of the trials that are ongoing right now are in the field of prostate cancer. Not because everyone, I think, wants to focus on prostate cancer. I do not. I like to focus on breast cancer. But because it worked so well, they have a target, which is PSMA on the prostate cancer cell, and binding agents, these prostate-specific membrane antigen binding agents that work so well. Once something works real well, more people want to build similar molecules so they can get into the field, right? So the PSMA has been so successful, that's why there's so many trials into it. And I think now the real push for the field is finding more targets that are as good as PSMA, building more binding agents that are as good as PSMA binding agents. Okay? So that's the future. In any case, for now, the PSMA on prostate cancer, we have all these different binding agents that are linked to radioisotopes like gallium, fluorine, copper for imaging, and then bound to lutetium, actinium, lead-212 for therapy. And this is a tremendous success in the field of prostate cancer research, where these PSMA-targeted imaging agents are able to detect disease to an extent that was never before possible. The sensitivity of these agents is far superior than anything that ever came before them. And the therapy agents are able to result in remarkable responses in some patients, right? Incredible efficacy with fewer side effects than the chemotherapies that would otherwise be used. So again, the prostate cancer patients is one patient group that we have not had much difficulty getting people to get onto clinical trials, because they would much rather have the radioligand therapies and their relatively gentle side effects than the chemotherapies with the much more severe side effects. This is the data that led to FDA approval of lutetium-177 PSMA 617, which is now Pluvicto. Very similar to what we talked about in the NETTER-1 trial for neuroendocrine tumors. The Pluvicto survival is a considerable improvement over prior standards of care, and so you have that efficacy as well as fewer side effects. You win-win. Now let's talk about how do we expand this from neuroendocrine and prostate to cancers of all types. Well, as we said, what we first need is to discover a good target and then build binding agents that hit that target. The easiest thing to do is to take a target that we already know about and binding agents that we already have and see if they are expressed in other tumor types. So this is some of the work that we did. This is the slide I showed for neuroendocrine tumor cells. However, it turns out the exact same target, somatostatin receptors, are expressed on some breast cancer cells. Not to get too technical, but about 50% of ER-positive breast cancer tumors express somatostatin receptors. So the exact same target that is utilized for neuroendocrine treatment may be able to be used for patients with ER-positive breast cancer. Well, first you have to show that they have the target. Okay? So I think people are usually familiar with FDG PET scans, right? So here are just a series of patients with FDG PET scans, disease seen on the FDG PET in all of these patients with ER-positive breast cancer. They underwent the somatostatin targeted PET scans, and in some, only about 30%, But in those 30%, we could see excellent uptake of these imaging agents in the breast cancer metastases. Now, this is not going to be an imaging agent for patients with breast cancer, because if you only see 30% of patients' disease, that's not good enough. But now I said we can swap out the radio tracer. So we can take those patients that we find the disease on that targets SSTR2, put in a radio tracer like actinium-225, which emits enough radiation to kill cancer cells, and this is the first patient treated with actinium dotatate who had an ER-positive breast cancer, where before therapy the disease was very prominent and the patient needed weekly thoracenteses to drain fluid from their chest cavity in order for her to breathe. And after 2 treatments, there's almost an entire resolution of the disease. Now, here's a warning. The first patient always works the best. Okay? I only wish that every time we had a success like this, it turns out that it works just as well on everyone. But this is a potential area of extending theranostics to patients with breast cancer. There's an ongoing clinical trial right now called the Tracy-1 trial, named after our patient whose first name was Tracy. I was very pleased that they did that. They named the trial after her. And this is an ongoing trial to evaluate what we know works well in neuroendocrine tumors. Can it also work well in the select percentage of breast cancer patients that also express this target? Okay, so how do you, and while I'm right there, there are clinical trials now for not just prostate cancer, neuroendocrine tumors, breast cancer, but there's trials for lung cancer, pancreatic cancer, gastroesophageal cancers. Virtually every type of cancer that is known to humans is now under clinical trials for radioligand therapies, with the hope that what we see in prostate and neuroendocrine will translate into successes for patients with other types of tumor types. So if you're interested in a clinical trial, how do you evaluate whether you should or should not participate? And that is the consent form. Okay? These consent forms are written to be readable. They're just too bloody long. Okay? So to me, it's always a combination of the consent form and the physician who's introducing you to the consent form, because I know when I bring the 40-page consent form into the patient, virtually no one, okay, maybe a few lawyers, read the entire consent form. But for most patients, you need that intermediary, the physician, to try and summarize what are the important pieces of the consent form. So you have listening to the physician and being able to read through the consent form. What I'm really happy with is that consent forms are evolving towards better states all the time. And now what I see is most new trials are coming out with what we call like summaries of the consent form on the first 2 or 3 pages that go over the real meat, and then they still have the extra 37 pages of other stuff, which is, again, written in they don't use complicated words, but it's still just too long. Okay? But the consent form, particularly if there's a summary in the beginning, you want to understand the consent form. And this is one of the consent forms that we have for a trial of fibroblast activating protein, targeted treatments for patients with breast cancer, pancreatic cancers, and lung cancers, because multiple different tumor types express this FAP in their stroma cells, so we can target multiple tumors with the same agent. These consent forms, the first page will give you the title of the study. It will tell you who the principal investigator is and who are the other people who are working on the trial and where it is being performed. And then it will go by section, at least all of these questions. What is the purpose? I am testing drug A, that in this disease. It will tell you what are the treatments that you will receive. And these consent forms and the protocols are written to be very accurate, and the protocols are written so the doctor has to follow the protocol, dot every I, cross every T, so that no one is playing mix about bake a cake any way they want to. They have to follow all the rules. So it will tell you exactly what are the study treatments that you're going to receive. Who's paying for the study, which is usually the sponsor of the trial. There are ISTs, industry-sponsored trials. Those are funded by the pharmaceutical company. And then there are IITs, investigator-initiated trials, for example, trials that I write myself. Those are either funded by pharmaceutical companies that I have requested to fund my trial, or a government agency like the NIH that is funding the trial. Do you have to join this study? The answer is always no. This is entirely voluntary. So you get to choose whether you participate or not, and the option is you always have the option of participating or not participating. It tells you what the potential benefits are. If it's a therapy, there's a potential benefit that the medicine will make your disease better. They can never, if anyone ever guarantees you that they're going to benefit you, that is not the person I will want to trust. Okay? So any discussion of potential benefits should be, I cannot guarantee anything, but here are some things that I may be able to help with. What are the side effects? That's probably the most important part of the consent form, because you want to know what you're signing up for. If it's an imaging agent, the diagnostic studies, again, the side effects are virtually nothing. But therapy studies can have very significant side effects, and you want to know, hey, there's a 1 to 2% chance I'm going to get cardiomyositis, right? These are important things for you to know before you sign up for a trial. Will you be paid to be in a study or not? And what do you have to pay for? A clinical trial often offers all the research components free to you, right? I would encourage that. If someone also says, hey, I have a clinical trial, you only have to pay $20,000 to be part of it, that is not the clinical trial you want to be in, okay? So you should not pay for any research component. There may be some standard care components like lab tests or imaging, which are billed to your insurance, and this section of the consent form will lay out what you should and should not expect. Who has the rights of the results to your studies? Usually the pharma companies that are sponsoring. And is your information kept confidential? The answer to that is always, we try our best. But as I said, more people have eyes on you when you're part of a clinical trial, so more people will see your medical records if you're part of a clinical trial. So if you're a type of person who wants the fewest number of people to see your medical records as possible, clinical trials may not be for you. And then what if I have questions? Number one, the principal investigator, who is usually the one explaining the clinical trial to you in my lab, it's always me or my partner, right, one of the physicians. But there should always be a physician that can answer your questions. And then there should always be a section on who the institutional review board is that is overseeing the research. So if you have a complaint or a concern about me or my team that's performing the trial, you don't have to complain to me, right? You don't want to complain to the person you want to complain about. You need someone who's overseeing the person that's running the trial that you can go directly to and voice your concerns. All right? How do you find the clinical trial that's right for you? Well, often these trials are brought to your attention by a physician or a nurse navigator who is part of that trial or has a colleague that's part of that trial. But sometimes you want more than just at your location. So if you're at Harvard, Harvard has tons of clinical trials, but they still don't have all of them, and you want to be aware of what's available to you in other locations. So one thing I recommend is something called, this is a website, nct.gov. This is the National Clinical Trials Database. You can type in the disease, multiple myeloma, and it will give you a list of all the different clinical this probably will be like trying to take a drink of water out of a fire hydrant, but it will give you all the different trials that are being run in that particular condition. So if you have, it's particularly valuable if you have something that is rarer, like Erdheim-Chester disease. There are only a few clinical trials that are running in that space, and you'll be able to see every single clinical trial that's being run in the United States on Erdheim-Chester disease with one search on nct.gov. Now, if you have breast cancer, again, it's going to be like trying to take a drink of water from a fire hydrant, but you can limit things smaller. I'm interested in ER-negative, HER2-positive metastatic breast cancer post-chemotherapy. Boom. You can narrow your search down to find the trials that are of interest to you. Now, for radiopharmaceutical trials, there is a website as part of the SNMMI, Society of Nuclear Medicine Molecular Imaging.org website, which you can find called the Global Radiopharmaceutical Trial Finder. If you go to this website, you can find the clinical trials that are involved radiopharmaceuticals like myself. I'm going to take a little, do I have a minute? I know I'm getting to the end of my time, but I do like to mention this because I find this very amusing. SNMMI, Society of Nuclear Medicine and Molecular Imaging, the society that I've just been elected to be the vice president elect. That's a mouthful. Why is it SNM? Well, the society used to be SNM, Society of Nuclear Medicine, which was much easier to say, shorter, easier to remember. Our problem was Google. Because if you put SNM in Google, you got something other than our organization. So this is the thing that you're going to remember from my talk, I know, right? But we changed the name to the Society of Nuclear Medicine and Molecular Imaging because now when you put SNMMI in Google, you get us. Right? Okay, so if I have, this is my clinic at the Hoag Family Cancer Institute. We have currently 30 clinical radiopharmaceutical, molecular imaging and therapy clinical trials running for patients with prostate cancer, neuroendocrine tumors, breast cancer, lung cancer, pancreatic cancer, gastroesophageal cancer. This is our space. 3 lead-lined treatment rooms, our hot lab for receiving, dispensing the radioisotopes. This is my home away from home, my office right here. The all-important radioactive bathroom that only patients get to use, so we have to leave the clinic and go across the hall. But if you're a patient, then you can use that restroom. And if you have any questions, both my name with my contact information and our clinic coordinator, Beth Thompson, is listed on this slide. And a few pictures of lovely Southern California. It is this pretty. And thank you so much and best wishes to you all.
Okay, any questions for Dr.? Yes, there's a couple over here.
Hold on, I'll bring the microphone over.
Okay,
Hold on.
thanks, Jan.
Yeah, sure. Mason.
Hi, thank you, Dr. Ulaner.
I know Mason, and Mason, you call me Gary.
Hi,
Right?
Gary. Okay, thank you. So earlier in your presentation, you mentioned phase 3 is generally a large population. Are there adjustments or considerations for diseases that have smaller populations? Like what's considered large for a rare disease and who determines that? Is there guidance? Thanks.
So people smarter than me determine that. Often statisticians are the ones who decide upon what we call the N, the number of participants that are needed in a trial in order to prove statistically that an agent is both safe and efficacious. And it is taken into account how rare a disease is. So something like, again, like Erdheim-Chester disease, some agents for these rarer disease types have been FDA approved after just phase 2 trials because they know that they don't have 600 patients in which to run a phase 3 clinical trial. Mason is as an advocate for the Lobular Breast Cancer Alliance, and I think there's a misconception out there that lobular breast cancer is a rare disease because lobular only is about 15% of breast cancers, but 15% of a large number is a very large number. Right? So if lobular breast cancer were its own disease, it would be about the 7th or 8th most common cancer in humans. So while people tend to think lobular breast cancer is a rarer subtype of breast, there are unfortunately a lot of patients with lobular breast cancer, and if we have yet to hit that magical, what is the first treatment that's being targeted just at lobular breast cancers, but I would fully expect a trial to get FDA approval will need to be a phase 3 in lobular breast.
I have a question. My name's Jan Manor. I'm with YourProstateCancer.help. I think a lot of the general public still thinks that clinical trials have placebo, and that, in my view, is one of the first walls that makes them not even want to consider it. In prostate cancer, for example, things are compared, and it has been for several years now, to standard of care. So no one in the recent trials, including the Xofigo trials, the Pluvicto trials, received placebo alone.
Absolutely.
Right. So we need to get that message out that if you are in a clinical trial, ask, because it's probably not a placebo group. It's probably a standard of care group, which means you probably get standard of care for free, for no charge.
For metastatic cancer, that is absolutely the case, because there's virtually no metastatic. Well, let's say there probably are some. There will be some instances where the rare tumor doesn't have an existing therapy. That is
Correct.
Not what we're talking about for 99.99% of
Correct.
oncology clinical trials. In those cases, the new agent is always being tested against a standard of care which has already been proven to be safe and efficacious. So you're receiving at least as good as the standard of care, plus the opportunity of getting something newer.
So I want to encourage people to ask. Don't assume that if you're in a trial, you're going to get placebo and nobody tells you. Thank you. Josh?
Hi. Great presentation, Gary. One, just after you sign an informed consent, you can always withdraw from a trial. So it is not a binding contract that keeps you into the end. At any point, you are permitted, and it's actually outlined in an informed consent, how you can withdraw from a trial, and that's really an important thing. Yes, you don't have to sign it, you don't have to join, but you can also exit the study as well. That's all.
Thank you. How much time do we have, Karen? Lunch is here. One more question, two more questions? Okay, I'll be right there. Hold on.
So going back to your phase 1, 2, 3, it's interesting. I received PRT, lutetium-177 dotatate, as part of the NETTER-2 trial, which was a phase 3 trial, stage 4, grade 3 NET. And I received the 4 doses, 200 millicuries each, 8 weeks apart, and I was so new I didn't even think about dosimetry or anything like that. But being here yesterday during some of the sessions, a number of speakers basically intimated that, well, it wasn't exactly random, but there was a lot of guesswork, and maybe the deciders on the dosage were a little overly conservative. So when you look at new RPT trials, I just wonder about the relationship between identifying the correct dosage and efficacy, and how do you think about the fact that the correct dosage, the correct amount of activity may vary by person, and you know, there may be underdosing, there may be overdosing. Just
How do you get the right dose of the radiopharmaceutical?
And I'm just wondering if deciding the dosage up front is really the right way of doing it.
But unfortunately, there's no other way. We like to say, right, democracy is the worst form of government except for all the others. So the same with the dosing. Choosing 200 millicuries is the worst way of choosing except for all the others. There is yet to be a clinical trial that has demonstrated that patient-specific dosing leads to better outcomes, which is why the standard of cares that the FDA have approved have yet to incorporate this type of patient-specific dosing. I look forward to the day when that is proven and then does get FDA approved. But we deal with with the level of knowledge that we have today.
Hi, my name is Jerry Francoeur, and I'm one of the PAB members. This sort of goes along with his. I had I'm a 2-time breast cancer survivor, and then my dog got cancer, and I took him to the UF Vet school because I wanted a veterinary oncologist. And what I learned there surprised me totally on the difference of treatment to animals and to humans. Animals are dosed, and they have less side effects. The FDA says humans can have the maximum tolerable dose. So to me, that makes and I was a physician's assistant in oncology, that makes no sense that we can dose the animals, but we can't dose the humans. And I do know there are some things starting up, it started last year, that they're looking at dosing for humans, but it's been around for animals for a long time.
I look forward to the day when we have the data that shows us that patient-specific doses improves patients, and then it will become widely utilized. And I have never treated a non-human animal, so I have no experience.
Okay, listen, thank you, Dr. Ulaner.
Thank you very much.
Appreciate it.